Good morning. Myself, Dr. Ragasudha, Head and Assistant Professor, Department of Microbiology, Government City College, welcomes you back to Protein Synthesis Part 2. The mechanism of protein synthesis. Translation is similar in prokaryotes and eukaryotes. Here, we will explore how translation occurs in E. coli, a representative prokaryote, and specify any difference between bacterial and eukaryotic translation. Protein synthesis can be divided into three stages, namely initiation, elongation, and termination. The main features of initiation steps are binding of mRNA to the ribosome, selection of initiation codon, and binding of charged tRNA bearing the first amino acid, namely methionine or formyl methionine. In elongation stage, there are two processes, joining two adjacent amino acids by peptide bond formation and moving the mRNA and the ribosome with respect to one another in order that each codon can be translated successively. This movement is called translocation. In the final stage of protein synthesis, the completed protein is dissociated from the synthetic machinery and the ribosomes are released to begin another cycle of synthesis. The direction of translocation is always from 5' to 3' direction. Every protein molecule has an amino terminus and a carboxyl terminus. The 5' terminus is a amino terminus while the 3' terminus is a carboxyl terminus, and the synthesis always begins at the amino terminus, that is at the 5' end. Let's begin with initiation. The initiation of protein synthesis begins with the formation of an initiation complex. In E. coli, this complex involves a small 30S ribosome, the mRNA template, and three initiation factors that help the ribosome assemble correctly. They are guanosine triphosphate or GTP that act as an energy source and a special initiator tRNA carrying N-formyl methionine. In the picture we can see the initiation stage, where before initiation how the tRNA with first amino acid and the largest ribosomal subunit, that is 50S subunit, and the smallest ribosomal subunit 30S subunit. The largest subunit and the small subunits of the ribosomes are not bound with each other before initiation. In initiation, translational complex forms and tRNA brings first amino acid in polypeptide chain to bind to the start codon of the mRNA. The start codon is usually AUG. The tRNA consists of an anticodon called UAC. The initiator tRNA interacts with the start codon AUG of the mRNA and carries a formylated methionine. Because of its involvement in initiation, formylated methionine is inserted at the beginning end terminus of every polypeptide chain synthesized by the E. coli. In E. coli, mRNA, a leader sequence upstream of the first AUG codon, called the Shine-Dalgarno sequence, which is also known as ribosomal binding site. The Shine-Dalgarno sequence is made up of nucleides, namely AGGAGG. It interacts through complementary base pairing with the rRNA molecules that compose the ribosome. The rRNA molecule comprises of an complementary sequence, namely UCC-UCC-A on its 30S ribosomal subunit. This interaction anchors the 30S ribosomal subunit at the correct location on the mRNA template. At this point, the 50S ribosomal subunit then binds to the initiation complex, forming an intact ribosome. That is how the initiation of translation takes place in E. coli. The next step in translation is elongation. In prokaryotes and eukaryotes, the basis of elongation of translation are the same. In E. coli, the binding of the 50S ribosomal subunit to produce the intact ribosome forms three functionally important ribosomal subunits. The A site or amino acyl site binds incoming charged amino acyl tRNAs. The P site or peptidyl site binds charged tRNAs carrying amino acids that have formed a peptide bond with the growing polypeptide chain but have not yet dissociated from its corresponding tRNA. The E site releases dissociated tRNAs so that they can be recharged with the free amino acids. There is one notable exception to this assembly line of tRNAs. During initiation complex formation, bacterial F, MET and tRNA or eukaryotic methionine tRNAi enters the P site directly without first entering the A site, providing a free A site ready to accept the tRNA corresponding to the first codon after the AUG. So, the binding of 50S ribosomal subunit forms three functionally important ribosomal sites, namely amino acyl site, peptidyl site and exit site or E site. Amino acyl site, otherwise known as A site, it is a site where the tRNA binds incoming charged amino acyl tRNAs. Whereas peptidyl site provides binding of charged tRNAs carrying amino acids that have formed peptide bonds with growing polypeptide chain, while the third site is the exit site where it releases the dissociated tRNAs. Movement of mRNA and ribosome with respect to each other is called translocation. The peptide bonds formed between the amino group of the amino acid at the A site and the carboxyl group amino acid in the P site. The peptide bonding is catalyzed by peptidyl transferase found integrated with the 50S ribosomal subunit. This process requires energy derived from GTP hydrolysis catalyzed by specific elongation factors. On an average, E. coli translocates approximately 200 amino acids per 10 seconds. Elongation proceeds with single codon movements of the ribosomes, each called a translocation event. During each translocation event, the charged tRNA enters at the A site, then shifts to the P site and then finally to the E site for removal. Ribosomal movements or steps are induced by conformational changes that advance the ribosomes by 3 bases in the 3' direction. Peptide bond formed between the amino group of the amino acid attached at the A site and the carboxyl group of the amino acid attached at the P site. The formation of each peptide bond is catalyzed by peptidyl transferase and RNA based ribozyme that is integrated into the 50S ribosomal subunit. The amino acid bound to the P site tRNA is also linked to the growing polypeptide chain. As the ribosome steps across the mRNA, the former P site tRNA enters the E site detached from the amino acid and is expelled. Several of the steps during elongation, including binding of a charged amino acyl tRNA to the A site and translocation, requires energy derived from GTP hydrolysis, which is catalyzed by specific elongation factor. And amazingly, E. coli translocates apparatus takes only 0.05 seconds to add each amino acid, meaning 200 amino acid proteins can be translocated in just 10 seconds. The final stage of translation is termination. Termination of translation occurs when a nonsense codon called UAA, UAG or UGA, also known as stop codons, are encountered for which there is no complementary tRNA. On aligning with the A site, these nonsense codons are recognized by the release factors in the prokaryotes and eukaryotes that result in P site amino acid detaching from its tRNA, releasing the newly made polypeptide. The small and large ribosomal subunits dissociate from the mRNA and from each other. They are recruited almost immediately into another translation initiation complex. In the picture, we can see all the three stages of translation, namely initiation, elongation, and termination. Before initiation, we can see a dissociated ribosome which is having a separate larger subunit and a smaller subunit. The smaller subunit recognizes the AUG or the start codon on the mRNA. Translational complex forms and tRNA brings the first amino acid in the polypeptide chain to bind to the start codon on the mRNA. This is followed by elongation. In elongation, the tRNAs bring amino acids one by one to add to the polypeptide chain. While in the final stage of translation called termination, release factor recognizes stop codons and translational complex dissociates and completed polypeptide chain is released. Thus, the complex is ready. The components of the translation are, that is ribosomes and mRNA, all are ready for another cycle. In prokaryotes, the process of transcription and translation occurs simultaneously in the cytoplasm, allowing for a rapid cellular response to an environmental cue. In eukaryotes, the transcription is localized to the nucleus and translation is localized to the cytoplasm, separating these processes and necessitating RNA processing for stability. In the picture, we can see prokaryotic cell wherein we can see the translation and transcription are going on simultaneously in the cytoplasmic region. Whereas in the second picture called B, we can see eukaryotic cell wherein transcription and translation are separated. Translation is in the cytoplasmic region and transcription is in the nucleus. Protein targeting, folding and modification. During and after translation, polypeptides may need to be modified before they are biologically active. Post-translational modifications include removal of translated signal sequence, short tails of amino acids that aid in directing a protein to a specific cellular components, proper folding of the polypeptide and association of multiple polypeptide subunits, often facilitated by chaperone proteins, into a distinct three-dimensional structure. And the third modification includes proteolytic processing of an inactive polypeptide to release an active protein component. And the fourth modification includes various chemical modifications like phosphorylation, methylation and glycosylation of an individual amino acid often occur in post-translational modifications. So as we sum up, the mechanism of translation includes three stages, namely initiation, elongation and termination. The main features of the initiation step are binding of mRNA to the ribosome, selection of an initiation codon called AUG, and binding of a charged tRNA bearing first amino acid, that is formulated methionine or methionine. The second step, elongation stage, has two processes. One is joining two adjacent amino acids by peptide bond formation, and the second process is moving the mRNA and the ribosome with respect to one another in order that each codon can be translated successively, which is known as translocation. The third stage of protein synthesis includes termination stage, the complete protein is dissociated from the synthetic machinery and the ribosomes are released to begin another cycle of synthesis. The released protein often undergoes some post-translational modifications like removal of translated signal sequences, then folding of the polypeptide and association of multiple polypeptide subunits with the help of chaperone proteins to form a three-dimensional structure or a functional structure, and proteolytic processing of an inactive polypeptide to release an active protein component, and various chemical modifications occur in the post-translational modifications. Thank you all.